The composition of biological communities is driven by ecological processes that act at both the species and genetic levels. In fact, the assessment of biological diversity across organizational levels, genes, species, communities, provides key insight into such processes. This leads to a key ecological question: to which extent species and genetic diversity vary in concordance across biological communities? Theory predicts that the strength of the correlation between species abundance and genetic diversity should be related to community age in close communities. Abundant species accumulate more genetic diversity over time than rare species. Here we have tested such predictions for continental communities using leaf beetles community along a latitudinal gradient in the Iberian Peninsula. Community age is essential from the degree of climate stability since the Last Glacial Maximum. Thus, we predict that if the congruence between species abundance distribution and species genetic diversity depends on community age, then higher congruence would be observed in localities where climate has been more stable since the Last Glacial Maximum. For each community, we built the species abundance distribution with the x-axis representing the rank from the most abundant to the least abundant species. We also built the species genetic diversity distribution with the x-axis representing the rank from the species with the highest genetic diversity to the one with the lowest genetic diversity. To quantify the congruence between species abundance and species genetic diversity distributions in each locality, we compute the Pearson correlation between the species ranks in each distribution. As expected, we observed large variabilities in the SAD-SGD rank congruence values, which range from Pearson R minus 0.25 to R 0.48. Such variability was expected due to different stability conditions since the LGM. This was confirmed by the correlation between SAD-SGD congruence and temperature stability since the LGM. Taken all together, this study shows that the effect of long-term climatic stability on biological communities can be detected in the SAD-SGD rank congruence. Therefore, we can conclude that the degree of congruence between SADs and SGDs could be used as a proxy for community stability. And in climatical stable localities, species abundance could be used as a proxy for genetic diversity.